for the day is DNA fingerprinting and DNA profiling. We will start with understanding properties of the human genome which are useful for this application, and then we will go to understand the original method of DNA fingerprinting and then how it has evolved to the modern technique of DNA profiling. So to start with the human genome, see, the human genome, as you all know, is 3.1 gigabase pairs distributed into 24 chromosomes, and after 20 or 30 years of genomic study, scientists know that today it is estimated to contain around 20,000 protein coding genes. Now, finding this out has been a great achievement of modern science. What is also very surprising is that this sequence, 20,000 genes approximately, is only 1.1 to 1.5% of the human genome. That means this is only 1.5 maximum of this one. And that means there is a large part of the human genome which is non-coding DNA. And there are various kinds of non-coding DNA. There are the introns, there are the pseudogenes, there are the transposon elements, there are various kinds of repetitive DNA, and our focus is on this last term, repetitive DNA. The repetitive DNA can be grouped into what are called interspersed repeat DNA and tandem repeat DNA. We are interested in the tandem repeat DNA. Tandemly repeated DNA. Let us look at what we mean by that. What I mean by tandemly repeat DNA is this. Let's say there is a sequence G, A, T, A, C, C. 6 nucleotides. Now this sequence, if it is present, let's say this is our chromosome 1, and if this chromosome, so this is our chromosome 1, that's the centromere, and if this sequence is present over here once, twice, thrice, 4, 5, 6, the same sequence GATACC, again GATACC, again GATACC, GATACC. If this sequence is present one after the other in a tandem array, repeated, that is a tandem repetitive DNA. And we find these kind of sequences all over the genome. The size may vary. The size may be 6 base pairs, it could be 3 base pairs, it could be 50 base pairs, it could be 75 base pairs. The number of repeats could vary from position to position. There could be 70 base pairs of a repeat in chromosome 1 and say 35 base pairs in chromosome 3 and then 150 base pairs in chromosome 7 and so on. But they are there. These are tandemly repetitive DNA. The tandemly repetitive DNA is largely grouped into what are called satellite DNA, write it off again. Into satellite DNA, which are very large repeats. You know, the repeat unit is more than 150 or 200 nucleotides long, and that is present for kilobase after kilobase. And where are they present? Usually near the centromeres of all the chromosomes and in a lot of the heterochromatin regions. That is satellite DNA. We are not really interested in satellite DNA. We are interested in the smaller versions, that are the mini satellites and the microsatellites. That's what we are interested in. The mini satellites, this repeat unit is somewhere between 10 and 100 base pairs. You'll find different books giving you a slightly different estimates, but this is the broad range, 10 and 100 base pairs, or 100 nucleotides, you could say it that way. Okay. Although this is in DNA, even then. And the number of repeats present at any given chromosomal position is, say, between 5 and 50 times. The microsatellites are between 1 and 10 nucleotides, much smaller, and they are also present between 5 and 50 times, usually at any given chromosomal position. These are our interest. The mini satellites, I must tell you, are also known as variable number of tandem repeats. Why? I'll explain in a minute. And the microsatellites are also known as STRs, or simple tandem repeats. We will start with the mini satellites because that was used to start with DNA fingerprinting, and then we'll go for the microsatellites, which have largely replaced the mini satellites in the modern evolved technique of DNA profiling. So how did it all start? This all started with a British scientist named Alec Jeffreys in the 1980s, and Jeffreys was working on human genome DNA when he found out that there were regions of the genomic DNA which had repeat sequences. So this is in the almost 40 years ago, and he found out there were repeat sequences which were present in many regions of the genome. And he began investigating more. He started with one and then began investigating more, and he found more and more. And then what they found out was simply stunning. They found that the number of repeats at a given chromosome position, let's say there is a 10 nucleotide repeat at chromosome number 1, and it is repeated in my chromosome number 1 15 times. So total 10 into 15, 150 nucleotide stretch. In your chromosome number 1, it is not repeated 15 times, it is repeated 17 times. So 10 into 17, 170 stretch. In a third person, your friend, it could be repeated 11 times. Not 15, not 17, but 11 times. So 110 nucleotide stretch. That meant this was varying from person to person. The number of mini satellites, the stretch, the number of repeats in the array was varying from person to person, and hence they were named, these mini satellites were named as variable number of tandem repeats. And Jeffreys realized that this he could use with the technique of southern hybridization to identify different people. Now, how did he go about that? It is basically this. Say this is chromosome number 1 of one person. All right. So here is a repeat unit whose size, I name it R1, and its size is 10 nucleotides. So this repeat unit is present over here once, twice, 3 times, 4, 5 times. In the homologous chromosome, it is present once, twice, thrice. So this is R1, locus R, repeat R1. There is another locus which is R2, not locus, sorry, another repeat R2, and the size of that is, let's say, 20 nucleotides. And it is present at a different position of chromosome 1. It is present 4 times over here, and in the homologous chromosome it is present 6 times. So this is 5, 3, 4, 6. Jeffries also realized that while the number of repeats was varying, the sequences flanking the repeat was constant for the chromosomes. And it was constant even between different individuals. So now what he did, he digested genomic DNA with restriction enzymes. So like HEY2 or HINF1, he ran them on an agarose gel. The bands separated as per their size. He transferred them to a membrane, and then he probed those membranes with a radioactively labeled short nuclear DNA, which was complementary to the repeat sequences, separately for R1, separately for R2. He had 2 probes. He probed for R1, for R2, and he got a set of bands. Now let us assume that the restriction sites cut in the region flanking the repeat DNA. So it cuts over here, it cuts over here, it cuts over here, 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 here. The gap in each case is 5 base pairs. Here, here, all 5. So what is the size of the fragment that we expect out of this? The fragment size will be 5 plus R1 is 10, so 50 plus 5, 60. For the homologous chromosome, same position, it will be 5 plus 30 plus 5, 40. For R2, for this individual, it is going to be 20, so 80 plus 5 plus 5, 90, and 1, 2, 3, 4, 5, 6, 120, and 5 plus 5, 130. So for the first person, for the first individual that we are testing, these are the size of the bands that will be picked up by the southern hybridization when he probed it with the probes which bind to R1 and R2. What Jeffries noticed was that if we did for another person, these numbers, the size of the fragments was varying because the number of bands present was different from person to person. So for this person, you got R1 giving you bands of size 60 base pairs and 40 base pairs. For another person, it could be, let's say, 70 base pairs and 50 base pairs. And for R2, it could be 100 base pairs and 140 base pairs. And for a third person, it could be, let's say, 50 base pairs and 80 base pairs. And for this person, it could become 80 base pairs and 120 base pairs. So the banding pattern was varying from person to person because the number of repeats present in tandem array at the same chromosomal position was varying from person to person. Now, if you consider these repeats as alleles, they were basically multi-allelic. Jeffries realized that this could now be used as he kept on building up more and more repeats and more probes. He realized that this could be used as a genetic fingerprint, because every person, the number of bands, the size of the bands that every person was giving was unique. In no case, if it was not happening for 2 sets of probes, it will happen for 5 sets of probes. You will pick up different size bands because you are looking at different mini satellites, and there is a variable number. There is the number of repeats. The number of repeats in a tandem array varies from person to person. So this, if you could look into quite a few probes, quite a few mini satellites, you will find differences from person to person, and this is unique for every person. That is why they named it as DNA fingerprint, as unique and rather more unique than the conventional fingerprint which is used for forensic analysis, for for signing on a stamp, for when people cannot sign, they put their mark, the fingerprint on the stamp paper for legal purposes and all that. Jeffries now realized that this could be used to identify different individuals, provided there was DNA. If you had DNA, if a crime had happened and the police were looking for the criminal, the murderer, let's say murder has happened, and the police are looking for the murderer, and there are a number of suspects, then they have to collect cells, tissue, something from the scene, crime scene, from which they can retrieve DNA. And if that DNA matches with any of the suspects, there is a very strong possibility that that suspect is the one who did the crime. And this can be presented in a court of law. Jeffries' technique was first used in the 1980s to settle an immigration case in the United Kingdom, and the very next year the police used it to solve a double murder case of 2 young girls who had been murdered, and the criminal was arrested based on the DNA fingerprinting evidence. Over the years, this became a very useful technique. More and more mini satellites were probed. They were used were identified, they were used as probes, and the method evolved. Different countries came up with standardized sets of probes. For example, the United States came up later on with what is known as the CODIS probes for profiling, of course. Okay. And these were used to identify, and large databases have been built up. In India, for example, you have one of the pioneering institutes, it's the Center for DNA Fingerprinting and Diagnostics in Hyderabad. There the various forensic departments use these studies so extensively, and it has evolved over the times. The only problem with this thing was that it needs a lot of DNA, because this is involving southern hybridization, and southern hybridization needs a lot of DNA. Otherwise, the probe will not find enough DNA to go and bind over there and give a signal. Plus you need intact DNA. So if you don't find a good amount of DNA from the crime scene, and if you don't find intact DNA could get fragmented, could get partially destroyed, all these things could happen. So then it becomes a bit difficult to do the analysis. And because it was also southern, the practical problem was that there was a difference in the intent. There could be differences in the intensity of the bands, and if that did not match properly with the DNA found from the crime site and between the suspects, the case could be argued that this is not the DNA is not of that of the suspect, so people could get acquitted even they were actually guilty. So this problem stayed on. Plus the usual problems that it is not possible to do automated southern, all these things came. That is when the technology began to shift to the microsatellites. Now, what are microsatellites? Also called as simple tandem repeats. These are smaller repeat units which are between 1 and 10 nucleotides long. Say it could be something like A, like this, dot, 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 dot. This is there. Or it could be something like CT, 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 sorry, my mistake, CT. Or it could be something like A, C, G, A, C, G, A, C, G, A, G. Or it could be a tetranucleotide like GATA, GATA, dot, 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 GATA. So the microsatellite repeat unit could be between 1 and 10 nucleotides, and they are also repeated, let's say, 5 and 50 times at different positions of the genome. And the microsatellites became very popular since the late 1990s for this kind of study because they are much smaller. That is why even if DNA was fragmented or partly damaged when it is retrieved from the crime scene, it could be used. They can be analyzed by PCR compared to Southern. I'll explain in a minute. And importantly, they are spread all over the genome. There are thousands and thousands of microsatellites present all over the genome, not restricted like mini satellites to centromeric position or subtelomeric position, and they could be used for this kind of analysis. The big thing also was that, as I said just now, that they could be used for PCR analysis, which is much more sensitive and much faster compared to the original Southern technique that Jeffries had used for the VNTRs, the mini satellites. So how does this work here? Basically, the idea is still very much the same. But here, instead of restriction enzymes, you use PCR primers. So let's say this is chromosome 1, and you have a microsatellite here which is being repeated 1, 2, 3, 4 times, and over here it is being repeated 1, 2, 3, 4, 5 times. And in another individual, I'm just taking one of the chromosomes here, it is being repeated 1, 2, 3, 4, 5, 6 times, and it is being repeated 1, 2, 3 times. Now, in the original DNA fingerprinting involving Southern, there will be restriction dilutions. You don't do that. Because now anymore in DNA profiling, they know that while the number of repeats, the number of microsatellites is varying, the flanking regions are constant for individuals. And so if you have a primer which anneals to this flanking region, then that will anneal to all different sources, different individuals' genomic DNA, and you can get a PCR product out of this, and out of this, and out of this, and this, but their sizes will vary because the size of the PCR product will depend on how many repeat units are over here. So for example, if the PCR product size over here is 50 base pairs, the PCR product size over here will be 70 base pairs. And the PCR product size over here will be 60 base pairs, let's say, and the PCR product over here will be 40 base pairs. And this I'm showing you only for one microsatellite or STR for one chromosome. Think about it, if you could build it up for all the chromosomes and use multiple primer sets for multiple mini satellite repeats. So within a very short time, you get different banding patterns, and they vary from person to person because the number of microsatellite repeats at any given chromosomal position usually varies from person to person. The chances that two unrelated individuals could have the same band, same set of bands is maybe 1 in 10 to the power 15 people or 10 to the power 19 people, which is much more than the human population of the planet. And so there is no chance, practically no chance of, making a mistaken identity. If sufficient DNA has been found from the site, it is going to work. That is the basis of the modern technique of DNA profiling. So this is what is today called DNA profiling, and you have different countries who have their standardized sets of probes and primer sets which are used for this kind of analysis, and they have been building with understanding how gender differences are found by this kind of DNA profiling. So whether a particular DNA comes from a female or from a male, how to identify that by this kind of study, and that involves the gene amylogenin. Now, amylogenin is a gene which codes for a protein found in the enamel of the teeth. The amylogenin gene is present on the X and the Y chromosomes. But the lucky thing for scientists is that the amylogenin gene on the X chromosome is shorter by 6 base pairs compared to the homologue on the Y chromosome. This is because in the X chromosome there is a 6 base pair deletion in one of the introns of the amylogenin gene. So this X chromosome amylogenin gene is shorter than the Y chromosome amylogenin homologue by 6 base pairs. That means, let us say, if you are doing a PCR and this is the amylogenin gene, and we are doing a PCR for profiling study, and you have the primers. This is the X chromosome and this is the same part of the Y chromosome. If we get, this is for the X chromosome and this is for the Y chromosome. If for this we get a product of 106 base pairs, a PCR product, for this we will get a PCR product of 112 base pairs, because the X chromosome amylogenin gene is shorter, right? And we have amplified part of the amylogenin gene which contains that difference. So for the X chromosome the product will be 106 base pairs, for the Y chromosome it will be 112. Now if you use these kind of primers on a set of for different people, for a DNA found from a crime site, if the DNA is from a female, what we will get is only one band, because both the Y chromosomes will give 106, 106. But if the DNA belongs to a male, we will get 106 from the X chromosome and 112 from the Y chromosome. And that is enough to tell us whether the DNA came from a male or a female, which becomes very important in many kinds of analysis. I'm sure you will agree. So remember this, amylogenin gene for gender identification in DNA profiling cases. And this is used in conjunction with all the with all the microsatellite analysis that is done. Thank you.